The overall goal of this procedure is to provide standardized conditions for successful seed sterilization of a large number of Arabidopsis genotypes while preventing microbial growth.
This method allows you to sterilize hundreds of seed samples at a time, thus simplifying a critical plant material preparation step in the fields of genomics, proteomics, metabolomics, and other high-throughput studies.
The main advantage of this technique is that it provides conditions in which efficient sterilization is combined with a high germination rate for a large number of seed samples.
To begin, prepare a 50% bleach solution with distilled water. To every 200 milliliters of bleach solution, add 50 microliters of tween 20 detergent. The bleach solution can be stored for up to a month as long as it is only opened in sterile conditions. Next, aliquot 100 seeds into a 1.5 milliliter microcentrifuge tube. This can be done manually or by using a custom-made seed robot. Now, in a laminar flow hood, add 500 microliters of the 50% bleach solution to the seeds. Then, transfer the tubes to a rotator and incubate the seeds in the bleach with mild agitation for 10 minutes. After 10 minutes, aspirate the bleach solution from the tubes, removing as much as possible. Then, add back 500 microliters of sterile distilled water and invert the tubes to mix. Allow the seeds to settle, then aspirate the water, and replace it with another aliquot to wash the seeds again. Perform a total of 6 such washes with sterilized distilled water.